for today's speaker, who will be Alessandro Marrero. Alessandro is a PhD researcher at the Institute of Biomedical Technologies at the University of La Laguna in Tenerife. He's a fellow from the Agencia Canaria de Investigación and is part of the CIBERNED group led by Abraham Acevedo. Alessandro is a graduate in biology from the University of La Laguna. And as I said, he's been carrying out his PhD, which is almost finished, as I understand, in Abraham Acevedo's laboratory. His scientific interest is basically around the functional interaction between TDP-43 and USP10, which is actually the focus of his talk today. So, Alessandro, thank you very much for your willingness to share your work with us today. And please go ahead. You can share the screen with your presentation and go ahead when you're ready. Thank you.
Perfect. Thank you and good afternoon. And okay. Okay. Good afternoon. My name is Alessandro Marrero Gallardi from the Abraham Acevedo's lab on the Unidad de Investigación del Hospital Universitario de Canarias. And today I am presenting our last result about the identification of a new functional interaction between TDP-43 and USP10. I have to say that all of this work is still unpublished, and these results are in collaboration with my colleague Judith Nodamayor. Okay, just to make a little introduction, I have to say that ALS are devastating neurodegenerative disorder that causing motor neuron or cognitive impairment. These two diseases, ALS and FTD, sharing genetic, neuropathologic, and clinical features. Both diseases are characterized by pathological protein inclusion in affected neurons, and almost all patients of ALS have this pathological inclusion of TDP-43 in upper and lower motor neuron, and approximately 45% of FTD patients present this inclusion of TDP-43 in the cytoplasm. Okay, TDP-43 is predominantly nuclear, where it has essential roles in splicing regulation and RNA metabolism. But in the disease, it is mislocalized and aggregate in the cytoplasm on affected neurons. This mislocalization leads to a loss of several nuclear function and also a gain of some toxic cytoplasmic function. There are some literature that show that ALS-associated RNA binding protein like TDP-43 are accumulated in stress granules during a prolonged stress. Stress granules are structures that contain stalled translation pre-initiation complexes that are assembled into discrete granules by specific RNA binding protein. Stress granules are implicated in neurodegenerative disease like ALS and FTD. So we thought that maybe modulation of stress granules could be critical to the pathologic aggregation of TDP-43. And there are several literature that show that USP10 modulate the stress granules formation by binding in an inhibitory manner to the stress granules formation complex. Also, recently, USP10 has begun to be related to some neurodegenerative processes like formation of tau-positive stress granules in aggresomes in Parkinson's disease, and in 2022 was related with aggregation of TDP-43. In this very recent article, show that USP10 is related with TDP-43, but in our research, this article shows same aspect similar to our TDP-43 aggregation data, but we also investigate the functional insights and implication of USP10 in the pathology. So for all of this, we have hypothesized that USP10 could be controlling the pathological aggregation process and the functionality of TDP-43. Okay. In our research, we use different cell lines like HeLa cell, U2S, or SH, SY, 5Y cells. And we have some models, but one of them is overexpression of GFP TDP-43 construct in wild type, and also with a mutation in the nuclear localization signal. So this TDP-43 delta NLS is cytoplasmic and is aggregate in the cytoplasm. We also use another model that is a stress-induced aggregation model of TDP-43, and with two models, we try to reproduce the TDP-43 proteinopathy characteristic of ALS and FTD, and then we test the effect produced by expression or knockdown of USP10. We also use the immortalized mouse embryonic fibroblast from knock-in TAR-DBP F210 mice with a partial loss of function. These mice have a mutation in the second RNA recognition motif, and there are partial loss of function produced by the inability of well union to TDP-43 to RNA. We also, in our research, we also use short harping RNA TAR-DBP doxycycline inducible cell lines that are HeLa cells and SH, SY5Y cells. And in the last part of our work, we also use ALS and FTD patient samples. Okay, we start our work studying the aggregation of TDP-43 and the role of USP10 in this process. And for that, we first overexpress the construct GFP TDP-43 delta NLS. And in this case, TDP-43 is aggregate in the cytoplasm. And we found that with the co-expression of USP10, USP10 inhibit the cytoplasmic aggregation of USP10 of TDP-43. Sorry. We repeat this experiment, but with the downregulation of USP10 instead to the overexpress of USP10, and in this case with the overexpression to TDP-43 delta NLS, we found that there are a lot of cytoplasmic inclusion, but with the knockdown of USP10, the size of this inclusion are bigger. So we can say that USP10 modulates the cytoplasmic aggregation of TDP-43. After that, we wanted to test if in this model, TDP-43 delta NLS inclusion are linked with stress granules. For that, we also overexpress the construct GFP TDP-43 delta NLS, but in this case, we also label the stress granules using a specific antibody against G3BP1. In this, we found that in this model, TDP-43 delta NLS inclusion are linked to the stress granules. After that, we also co-express USP10, and we found that USP10 inhibit the formation of these positive TDP-43 positive stress granules. So it seems that, at least in this model, USP10 modulate TDP-43 aggregation by inhibiting stress granules formation. To test that, we repeat the overexpression of TDP-43 delta NLS, forming this cytoplasmic inclusion, and we treat with a drug that it is emitting, that this drug block the stress granules assembly, and by inhibiting the formation of stress granules with this drug that it is emitting, TDP-43 is not able to aggregate in the cytoplasm. So it seems that this effect is dependent of stress granules formation. We wanted to test that, and in this case, we also use some mutant of USP10. Another time we overexpress the TDP-43 delta NLS to reproduce this TDP-43 proteinopathy. USP10 inhibit this cytoplasmic aggregation of TDP-43, and After that, we overexpress with this mutant that is USP10 F10A, that it is a mutant that doesn't bind to G3BP1. G3BP1 is a central protein a core protein in the formation of the stress granules, and this USP10 is unable to bind to G3BP1 and unable to interact with the stress granules. And we found that with this mutant of USP10, this USP10 is unable to inhibit the aggregation of TDP-43. So we can say that USP10 modulate the aggregation of TDP-43 through its interaction with G3BP1. After that, we also test another mutant of USP10. In this case, is C424A mutant, that it is enzymatically inactive, that don't have is catalytic deubiquitase inactive. So in this case, this USP10 mutant have the same effect to the wild type. So we can say that USP10 modulate the aggregation of TDP-43 through its interaction with G3BP, but independently of its catalytic deubiquitinase activity. After that, we also wanted to test the effect of USP10 in the aggregation of TDP-43, but using another model, in this case without the overexpression of TDP-43. And in this case, we use this stress-induced model. We stress the cells with the oxidative and osmotic stress using sodium arsenide and sorbitol, and the cell form this TDP-43 inclusion, cytoplasmic inclusion, and also these stress granules. And in this model that it is with endogenous TDP, there are not overexpressed TDP. And also in this model, USP10 is able to inhibit the cytoplasmic aggregation of TDP-43. Okay. After that, we also want to test if USP10 are also controlling not only the aggregation, but also controlling the expression of TDP-43. And we start to make some experiment of immunoprecipitation to test if TDP-43 and USP10 co-immunoprecipitate, and we found that yes, TDP-43 and USP10 co-immunoprecipitate, and we demonstrate that this interaction there are a direct interaction. With this data, we wanted to test if USP10 is able to modify the protein levels of TDP-43. To test that, we first overexpress USP10 and test the endogenous level of TDP-43. In this case, it seems that USP10 don't modify the endogenous level of TDP-43, but TDP-43 have a characteristic autoregulation processes. And we thought that maybe these autoregulation processes would be implicated. So in this case, we use also an overexpressed TDP-43 that is unable to autoregulate because this construct haven't a 3' UTR region that is necessary to this autoregulation process. And when we use this overexpressed TDP-43, when we overexpress USP10 increase the level of TDP-43. So we thought that we think that USP10 maybe controls the level of TDP-43 because we see it in this overexpressed construct. After that, we check this experiment, but with the knockdown of USP10 instead of the overexpress of USP10, and we have the similar data. When we downregulate USP10, the levels of endogenous TDP-43 there are not change, but when we test the level of overexpressed TDP-43 unable to autoregulate, we found that the knockdown of USP10 decreased the level of TDP-43. So we can say that USP10 control the level of TDP-43 protein. Okay. With all of this data, we wanted to know if this modulation of TDP-43 by USP10 have also a functional consequence, and if USP10 could be modulating the functionality of TDP-43. Briefly, I have to say that TDP-43 is an RNA binding protein. It has essential roles in RNA metabolism, controlling splicing of 100 exons. And recent data suggests that TDP-43 loss of splicing function might be critical for disease end-stage toxicity. And data from our lab also demonstrate that pathological mutation in C-terminal domain of TDP-43 leads to a gain of splicing function. All of these show the importance of TDP-43 functionality in pathology. So we wanted to test if USP10 are modulating the TDP-43 function. To do that, we use the CFTR splicing assay that it consists in CFTR is a TDP-43 splicing target. We use CFTR minigene, widely used as an indicator of TDP-43 splicing function. TDP-43 control the inclusion or exclusion of the exon 9 of CFTR. And we do this by the RT-PCR with specific primer against CFTR minigene and quantification the exon 9 exclusion as a functionality reporter of TDP-43. So in this experiment, we quantify the exclusion of exon 9, and we found that when we overexpress USP10, the USP10 can enhance the TDP-43 splicing function. And in this case, USP10 increased the splicing function of endogenous TDP-43 without modifying its protein levels, because in the previous data, I showed that in the endogenous, TDP-43 don't change its level, and USP10 can increase the splicing function without modifying its protein levels. After that, we also want to test that with the overexpressed different construct of TDP-43. In this case, we start to overexpress TDP-43 delta NLS, that it is cytoplasmic and aggregate in the cytoplasm, and there are a loss of function, and the overexpression of USP10 can rescue this functionality of TDP-43. We also overexpress the TDP-43 wild type that obviously enhance the splicing function of TDP-43, but when we co-express USP10, this activity is enhanced. So USP10 increase the splicing function of TDP-43. With this data, after we have this data, we wanted to know the possible mechanism by which USP10 is modulating TDP-43 function. And for that, we also use not only the previous construct, but also the construct of USP10, the overexpression of USP10 with different mutation. In this case, we first overexpress TDP-43 delta NLS, and there are a loss of function. USP10 wild type can rescue this functionality. USP10 C424A, that it is a USP10 mutant without the catalytic dehubiquitinase activity, is unable, it has the same effect to the wild type. So we can say that the effect of USP10 on TDP splicing function are not mediated by the USP10 dehubiquitinase activity. But surprisingly, this USP10 F10A mutant, that is the USP10 mutant that is unable to bind to G3BP1, this mutant is unable to rescue the functionality of TDP-43. So we can say that the effect of USP10 on TDP-43 splicing function is mediated by USP10 interaction with G3BP. Okay, which all of these data in which USP10 can control some aspect of TDP-43 biology and after demonstrate the direct interaction between TDP-43 and USP10, we wanted to test if not only USP10 modulate TDP-43, but if also TDP-43 are modulating USP10 expression. To do that, we overexpress TDP-43, wild type and delta NLS, that is cytoplasmic aggregated, and we found that we quantified the levels of protein levels of USP10, and we found that the overexpression, the TDP-43, led to decrease in USP10 protein levels. So it seemed that also TDP-43 controls the USP10 protein levels. We want to test this result, but in this case with a TDP-43 downregulation. In this case, we use HeLa cells, short hairpin RNA TAR-DBP, doxycycline inducible, in a collaboration with Manuel Portero team and Pascual Torres that have sent us to these cell lines. And we repeat this result with SH, SY5Y cells in collaboration with Pietro Fratta's lab. And we found that when we downregulate progressive TDP-43, the levels of USP10 go also down in protein levels, but only when in the mRNA levels. So it seems that not only USP10 controls the TDP-43 protein levels, but also TDP-43 are controlling the USP10 expression. Okay. With this previous data, in which TDP-43 could modulate in USP10 expression, we wanted to approximate to the pathology and test if dysfunctional of pathological condition of TDP-43 are modifying the levels of USP10. To study that, we use this knock-in mouse model. This knock-in mouse model that is immortalized mouse embryonic fibroblasts from knock-in mice with this hypomorphic allele in TAR-DBP F210I that have a partial loss of function of TDP-43 by the diminished RNA binding activity of TDP-43. These knock-in model are unique, and we use that like a chronicle model of a loss of function of TDP-43. And in this case, we found that with this loss of function of TDP-43, the levels of USP10 are significantly increased. Protein levels, but also the mRNA levels. So it seems that in dysfunctional condition of TDP-43, USP10 increase its levels. And with this data, that in dysfunctional condition of TDP-43, we found that USP10 levels are increased. So we asked ourselves if also in pathological condition of TDP-43, with this potentially loss of function, USP10 level would also be increased. To test that, I had the opportunity to stay for 3 months at the Institute of Neurology of University College of London, in Pietro Fratta's lab, and we were able to get the samples thanks to the pathologist Leslie Tamarin. And with these frontal cortex samples from FTD patient, we found that the levels of USP10 are also increased, and especially are increased in patient with TDP-43 pathology type B. After that, we also wanted to repeat this experiment in patient from ALS, and in this case, in collaboration with Manuel Portero team and Pascual Torres, and the lysate that sent us the lysate from motor cortex of ALS patient. thanks to Isidro Ferrer. We also found that in motor cortex of ALS patient, the levels of USP10 are increased. And I have to say, and we are waiting for samples from spinal cord patient from the CM Foundation, just to test or to repeat this Western blot experiment also in spinal cord from patient. Okay, after that, in my stay in London and in collaboration with Leslie Tamarin and Alex Bampton in the brain bank of the UCL, that they helped me to interpret this result. We also made an immunohistochemistry experiment. So we found that USP10 is accumulated in cytoplasm of neuron from FTD patient with TDP-43 pathology type B. We repeat this experiment, but also in immunofluorescence, and we had the same result, that USP10 is accumulated in the cytoplasm of neurons from patient of FTD, especially in patient with TDP-43 pathology type B. These images are better, and there are some inclusion of or accumulate of USP10 in the cytoplasm of neurons from FTD patient. And there are our recent work that I can say that our conclusion are USP10 modulate the cytoplasmic aggregation and rescue the functionality of TDP-43, and it do so by binding to G3BP1 and inhibiting the stress granules formation, and independently of its catalytic activity. USP10 and TDP-43 mutually affect their protein levels, and USP10 increase its expression and modify its subcellular location in pathological condition of TDP-43. Thank you all for your attention, and my acknowledge to all of these people that helped me with our work and all of these collaborators. Thank you.
All right, thank you very much, Alessandro, for a nice presentation. So now the paper is open for discussion. As I say at the beginning, if anyone is interested in making a question, please send me a message through the chat option that you have in the upper part of your screen, and just stating your name and your affiliation, and I will give you the floor to make the question yourself. So there is no need for you to write down the question, just stating your name and affiliation. So while we wait for some questions, let me remind all of you that CIBERNED Webinar Series are held every Thursday, on Thursdays every other week at 3:30 p.m. We have posted already in the CIBERNED webpage the list of webinars for the next few months. So if you're interested in looking at future webinars, please go to CIBERNED webpage and check the next webinars until July, I believe is the list is already posted. So, okay, so having said that, again, if you are interested in making a question to Alessandro, please do so by sending a message through the chat option, stating your name and your affiliation, and we'll give you the floor. I don't see any questions yet. No questions? Anyone? I see a hand raised by Teresa Iglesias. Yes, Teresa, please go ahead.
Hello, and thanks very much for your talk. Do you hear me properly?
Yeah.
Okay, no, very nice findings and talk. Thank you. Are you thinking about using this UP10 as a therapeutic approach for this disease?
Yeah, sure. But we first would like to understand if USP10 have because in this dysfunctional and pathological condition of TDP-43, USP10 are upregulated, and we would like first to understand if the role of USP10 in the pathology, but that if it's maybe protective or not, and we have to test that first. But sure, we understand that USP10 can control in TDP-43 functionality and aggregation, and that maybe would be interesting to the pathology, to the pathology approximation. And
yes, so maybe you are thinking about just generating mice without this protein in the background of your disease.
Sorry, I can't hear very well.
I'm sorry, maybe it's probably my computer. I don't have it very. If you are thinking about making a mouse deficient in this protein, you know, in a model of ALS, so that you can see how it affects the disease in the mouse first.
Yeah, sure. It would be a very good mice model with without USP10. Yeah, sure.
And do you know if this mouse exists? So do you have the knockout for this available from somebody? No.
I think that I am not read any literature about that. I think that no.
Okay. No, thank you very much. Very nice work. Thank you.
Okay, thank you. Thank you.
All right, thank you, Teresa and Alessandro. We have another request from Jose Lucas.
Hi, can you hear me?
Yes.
Okay, thank you for your presentation. And I was wondering whether the ubiquitin hydroxylase activity of USP10, have you characterized it in detail? How relevant it is for the effect that you are seeing? The ubiquitin, can you hear me?
Yeah, now better.
Okay, whether the ubiquitin hydroxylase activity of USP10, how relevant can it be to your system? Have you explored this, or in general, has it been explored in ALS?
Yeah, I think that yes, it is tested, but I don't understand really well your question.
Because USP10, it's a protein that is able to hydrolyze ubiquitin chains, right?
Yeah.
So is that function important for the role you are seeing in the stress granule formation, sequestration, and so on?
Okay. No, that in our model, this we use for answer this question. We use this two mutant model that without the ubiquitinase activity, and this mutant don't do the work like a wild type. So we found that for degradation of stress granules inhibition of the formation of stress granules is not necessary, is not mediated by the ubiquitinase activity of USP10, but it is important the other main functionality of USP10, that it is the G3BP1 bind and its interaction with stress granules. And for disassembly, the aggregation, the pathological aggregation of TDP-43, at least in our model, it is critical, it is necessary, this binding to G3BP1.
But I was wondering whether the ubiquitin hydroxylase activity has been characterized in other aspects of the disease, since there are protein aggregates in neurodegeneration in general, whether this, I don't really follow the field in ALS, and I didn't know whether there were reports of USP ubiquitinating activity in the formation of aggregates in ALS in general.
No, I think that it is not, al menos, at least it's not published anything like that.
Okay. Thank you
Okay.
for your results.
Thank you.
All right. Thank you, Jose and Alessandro. So any other questions? I don't see any other question coming, but if anyone is still interested in making a question, this is your last chance. Please either raise your hand or send a message through the chat option. Okay, so it appears there are no more questions. So then I think we are gonna close the session here. Not before thanking Alessandro for his willingness to share with us unpublished data, actually, and wishing him well for finishing his PhD in the next few months, and to thank everyone for attending the webinar, and I'll see you all in 2 weeks' time for the next one. All right. So thank you, everyone, and have a great day. Bye-bye.
Thank you. Thank you. Bye.